In October of 2020, Jennifer Doudna and Emmanuelle Charpentier won the Nobel Prize in Chemistry. They invented a revolutionary technique to make changes to DNA called CRISPR-Cas9. This technique can find specific regions in the human genome and make changes to the DNA sequence, potentially treating and preventing diseases. CRISPR is far superior to any other DNA editing technique because of its far greater specificity and efficacy in changing DNA. So far, 33,000 papers describing CRISPR have been published. Most of these use CRISPR in a laboratory setting. However, CRISPR has an amazing potential to be used in the clinic. Approximately 6 out of 10 people are affected by hereditary diseases. That means that 4.6 billion people worldwide have some form of hereditary disease. These diseases are caused by unwanted changes in our DNA sequence that disrupt the normal functioning of our genes. Many of these diseases are monogenetic diseases, in which only one gene in our genome is affected. Cystic fibrosis, sickle cell anemia, and Duchenne muscular dystrophy are all examples of monogenetic diseases. Patients with monogenetic diseases require complex treatments, often involving many different medications, leading to an average of $22,000 per year in individual treatment costs. Instead of treating individual symptoms, CRISPR could be used to restore the underlying DNA defects to create a healthy gene. With this one treatment, CRISPR can completely heal patients with hereditary disease, eliminating the need for a lifetime of medications. In addition to existing patients, CRISPR can be used to restore DNA defects in unborn babies. Using CRISPR, human DNA can be optimized to be perfect before we're even born. Taking evolution into our own hands, these so-called designer babies could be the start of a perfect human generation. If CRISPR is so promising, then why aren't we already using it on humans? Despite all the positive effects of CRISPR, there are also a lot of challenges. Next to the DNA we want to target, CRISPR also has the tendency to make cuts in other parts of the genome, leading to unwanted DNA changes in other genes. These so-called off-target effects could destroy other genes, leading to development of even more diseases. So researchers first need to optimize the CRISPR technique in order to minimize the off-target effects before it can be used in a clinical setting. At this point, a total of 68 Phase 1 clinical trials using CRISPR have been concluded, with many more to come. However, the treatment remains very risky at best, and caution is advised. In 2018, Chinese researcher He Jiankui announced that he had used CRISPR on unborn twin babies. Although the babies were born healthy, this news was critically received by the scientific community. Scientists are convinced that it's too early and unethical to be using CRISPR on babies, and Jiankui received widespread condemnation from the global scientific community. He was fired from his university position and had to spend 3 years in prison for violating the scientific ethical code. Using CRISPR on babies could have an enormous impact on social life as well. Every parent would choose to change their baby's DNA in order to create a handsome, smart, athletic baby using expensive CRISPR techniques. This could create an even bigger disparity between rich people who can afford CRISPR treatment and poor people who cannot. Many researchers want to avoid this dystopian future. It's clear that there will be many challenges before we can use CRISPR in humans. Researchers need to refine the technique, and politicians need to lay out legislation for genome editing. When this is done, billions of people worldwide can enjoy CRISPR to treat hereditary diseases from their genomes. Like the rise of vaccines, CRISPR will lead to a new wave of disease extinction. But the question remains, if we can use CRISPR to cure disease, can we use it to enhance human biology?